Hi everybody. So I decided to record this video based on the issues that we experienced in one of the previous tutorials, but also because at this level of the course in Biology 234, this is what many students struggle with. So I think that it is a good idea that you can just go back to this as a point of reference. And we were talking about the Dutch elm in the first question of the first part of last week, tutorial number 9, I believe, where they talk about this species of tree, which is called a Dutch elm, and they tell us that it's susceptible to a fungus. We have two varieties of this tree, and one of them is a breed that is resistant to this particular fungus, but we know that this is located into a single gene, and that the allele that this breed Jefferson has is recessive relative to the non-resistant allele for that gene. So they tell us as well that we have identified some molecular markers located in different chromosomes along the DNA that are unique for each of these two breeds. So there is one unique for the Jefferson and there is one unique for the Pioneer. So let's look first at how the pure breeds of the elements, the individuals of our parental cross look like. So here we have the Jefferson. This is the representation of how a cell, a somatic cell of an individual of the Jefferson breed looks like. Here we can see that the gene for the resistance, the nomenclature that I'm using is res for the resistant gene, and there are two versions, two different alleles. So one is the Y, that is the one for resistance, and this one is going to be recessive to the dominant allele, which is going to be the wild type that is non-resistant. So those are going to be the two different quotations that I use. So this Jefferson individual is represented here as a diploid organism. You notice there are two copies of the gene represented in the two homolog chromosomes. One was inherited from dad, the other one was inherited from the mom. And here on the right we have the other breed, which is Pioneer. As you see, these are also pure breeds, so they are homozygous for the dominant allele. They also give us the information about the SSLPs. An SSLP is a molecular marker, and for the purposes of the course, you should just consider it as being different versions of the same molecular marker. An SSLP is formed by tandems, and what it is just the sequence of a series of nucleotides that are repeated multiple times along the DNA. How many times determines the length in nucleotides of that particular SSLP, and at the end of the day it gives us a relationship of how heavy the molecule is going to be. So let's look at an example. Here I have three different elements, three different versions of the same SSLP, the one located in whatever chromosome. And the elements that are repeated in tandem are going to be these ones, the series CAT. Here we have our 5 to 3' N, which is the direction in which we always read the genetic material, and the number of copies is what determines the name for this particular allele. So being highly polymorphic, we can have many different varieties of an SSLP. But the moment where it is important to, but something that is important for us to consider is that the highest the number of tandems, the larger the molecule gets as a whole. Therefore, the size of the molecule gets bigger. The bigger the molecule, the closest is gonna be from the well after in our electrophoretic gel. So if you recall the electrophoretic gel, in the electrophoretic gel, these are usually individuals, and there are usually two bands. Okay, where we have homozygote individuals, the two bands are together, making it look like a one thicker band. So the way that electrophoresis works is by carrying the DNA, which is a negative molecule, down the lane on a specific amount of time based on its migration following the cathode that is connected here. So it's basically like if someone was pulling the molecule. The heavier it is, the closest it's going to be to the top. So the longer the molecule, the closer, the higher it's going to be. And we don't really care about the real size of these molecules other than the size that they have relative to each other. In other words, we care about this one, which is the highest, the heaviest, being higher in the gel relative to this one below, and then this one, and this one being higher than this one, who is the lightest. Okay, so that's how you read and interpret the table that was given to you in the example. So here I am presenting to you the result of the cross between the Jefferson and the Pioneer individuals. And here I'm presenting to you as well, again, these are the cells, but now I'm representing also the SSLP marker in a different chromosome than the gene of interest. And if each of these pure breeds has a different molecular marker. So this one has 2 nucleotides and this one has 4 nucleotides. So again, this could represent 200 nucleotides in order to really to make apparent make evident the separation in the gel. But yeah, the only thing that we care about is that the 2 should be lower than the 4. Okay, and Jefferson has is a pure breed with SSLP2, and Pioneer is a pure breed with the SSLP4 marker. When we cross this is going to be called our parental cross. We end up with an F1 generation. And that F1 generation is going to be a dihybrid. That dihybrid is going to carry 1 copy of each of the parentals, 1 copy from the Pioneer, 1 copy from the Jefferson for each of the genes, but also it's going to be in heterozygosity for both of those genes. So the phenotype that this hybrid F1 is going to have is going to be non-resistant. And this was based on information that was given to us. The non-resistant phenotype is dominant over the resistance. This is traditional phenotypic expression. But then something different happens with the SSLP relationships. Because the truth is, and I asked some of you during the tutorial, is that this is not an example of codominance. Some people like to talk about this, like which of the 2 molecular markers is going to be dominant against the other, or which one is going to be recessive, or are they both going to be codominants? And the answer is that you are always going to have both bands in your gel, right? So that relationship of codominance, I mean, technically it would be codominance, but at this level of expression, we're talking about molecules, this doesn't really apply anymore. You are always going to have represented both of the contribution of each of the two alleles, right? If they end up being the same, like in the case of Jefferson or Pioneer, still you would get a double band, making obvious that both copies of the gene have the same weight or the same allele. So here we have our dihybrid. And that's how it's gonna look like, right? So let's prove that our res gene is in a different chromosome than our SSLP marker. In order to do that, we're gonna use a test cross, and this is something that many students struggle as well with. Whether to use a test cross or whether to cross this with a tester. This is again our dihybrid individual, representative of the F1, and the question is, whether or not we cross this with a tester to make a test cross, or if we cross this with another F1 in order to get an F2. The goal here is to analyze the gametic contributions that this F1 individual can produce based on their categorization between parental and recombinant gametes. So I'm gonna first try doing it by doing a test cross, which is what I recommend you to do. It's much simpler. So here, the first thing that we need to do is to identify what are the different gametes that this F1 individual can produce. And we can use our now familiar formula of 2 to the n, where 2 represents a gene with 2 different alleles, which is what we can observe here, right? So there are 2 different alleles. And how many genes are there in your organism with 2 different alleles? And that is 2, right? One for SSLP, one for RES. And that gives us a total of 4 gametes. If you are uncertain, you can make, I introduced you this that is called a decision tree in programming jargon. And it's very useful because it gives you the relationship of combinatorial, it gives you all the possible results out of a combinatorial problem, right? Where you start with one allele or another for the first gene, and then you combine them with the second allele. So that's a way to confirm which gametes you're actually dealing with. So these are the 4 different gametes. The next thing that I would like you to notice is that which ones are parentals and which ones are recombinants. It's usually a good idea to identify them as soon as possible. So here in my diagram, I drew them in such a way that the contribution of the chromosomes on the left side are those given by one of the parents, the Jefferson parent, and those on the right are those given by the contribution of the pioneer parent. So if you notice here, res Y and S goes with SSLP2, and res N goes with SSLP4. Those are our parental gametes, right? And the reason why we call them parental is because in the F1 individual inherited them as they are, like this, from their parents, right? This one came from the dad, who was maybe Jefferson, and this one from the mom, who might have been Pioneer. Of course, this can be the opposite, right? Just keep that in mind. Okay, so if res Y and SSLP2 go together, and those are the parentals, that means that this gamete here is parental. And if res N and SSLP4 go together, that means that this gamete here is also parental, which means that the other 2 must be recombinants. Combinations of genes, in this case the gene and the marker of interest, that weren't inherited, that weren't given to the F1 by their parents. So something that many students struggle a lot with was when they try to identify parentals and recombinants directly from an offspring or from from a given table. So the truth, and that ended up being messy because some of you ended up calculating 3, 4, or even 6 different recombinants and only 2 parentals. When you're dealing with an F1, when you're dealing with a dihybrid organism where there are 4 different types of gametes, there is always going to be only 2 that are parental and only 2 that are recombinant. Okay. As the 2 genes get closer, the 2 genes, when the gene and the marker get closer and closer together, the frequency of the parentals goes up and up and up to the point when you might even consider, you might have the recombinants disappearing almost, right? So I'm gonna let you think about that one. Anyways, so we cross this. These are the contributions, the gametic contributions of our F1 individual, and then we cross this with the Jefferson parent. Why Jefferson? Because that is the homozygote recessive individual, the purebred for the gene of interest. And the only gamete that parent can produce is Res-Y and SSLP2, which is represented here. And then we just fill up our Punnett square. So we're going to have a direct relationship between the phenotype of the offspring and what the contribution of the F1 individual was. The first thing that we can do is to split the data into the things that we can actually see. The first thing that we can see is that there is half of the offspring that is resistant, and there is half of the offspring that is non-resistant. And from each of them, there is two different genotypes, one that is parental and one that is recombinant. So let's look at the resistant offspring first. Here we have again the two different genotypes that in which we can find the resistant trees. So if you notice, now that we extract this information, now that we used it to separate between resistant and non-resistant trees, we're just gonna focus instead on the information on the markers. We know that the contribution of one of the F1, the gametic contribution of the F1 with Y, and 2, the SSLP2, is the parental, right? That's how it looks like in the Jefferson. The Jefferson parent can produce Res-Y, SSLP2. Whereas this one and this one is a recombinant. The gamete that was passed by the F1 individual looked like this, which wasn't part of the Jefferson parent, right? So this happened in the meiosis during recombination in the gametes of the F1 individual. Okay, so this is gonna be two bands that are together, and this is gonna be two bands that are separated from each other, and this is how we look at them. So they all of the individuals, regardless of whether they're parentals or recombinants, have the same SSLP2, right? That is a constant. What changes is whether or not the other copy, that one given by the F1 individual, is SSLP4 or SSLP2 as well. So by identifying which one is parental and which one is recombinant, we can just count them. So this is going to be the parentals. These ones right here are going to be parentals, and these ones are going to be the recombinants. We count the recombinants. Here we have 14 is the number of wells. These are called wells. There are 7 out of 14. So that's 7 out of 14 that are recombinant. And then we look at the next gel. The one that belongs to that offspring that has trees that are non-resistant. And we do the same procedure. Which gamete belongs to the non-resistance? In this case was that of Pioneer, and Pioneer carried the SSLP4, right, which was this one. So that one was for Pioneer. So anything that has this in terms of the contribution of the F1 parent must be parental, whereas this one is the recombinant. If you do your graph again, your representation, you're gonna get a very similar pattern, but now it's gonna be inverted, right? Where these ones are gonna be the parentals, and these ones are gonna be the recombinants. So we count the recombinants again, and it's again 7 out of 14 that are recombinant. What is left to do is just to add up the information from the recombinants of both parents and divided by the total number of individuals, sorry, not both parents, like both phenotypes, and then divided by the total number of individuals in both wells, which is 28, right? There are 14 on each. And that is going to give us the final answer, which is the frequency of recombination, which is what we call map units, is 50 map units. Okay, and from here we can conclude that the gene of interest and the molecular markers are in different chromosomes. So something important to consider here is that whenever you are giving a problem like this, it is important to explain and identify which ones are parentals and which ones are recombinants. If you encounter this in a midterm or a final, it is important that you can tell us which one is recombinant and which one is parental in each of the gels that we're providing to you. Okay, you can very easily just count and then give us an answer, but forget to tell us what is what, and that's where many people lose marks. The other question that I get a lot is why can't we use only one of the gels? If they both give us 0.5, right? Like this gives us 7 out of 14 equals 0.5, and this one gives us 7 out of 14, which is 0.5 as well. And the reason why is because of the sample size. It is true that, you know, in a perfect world where your sample is perfectly randomized, like there is a population of individuals that are resistant trees, right? And you know that out of the population, 50% are going to be recombinant and 50% are going to be parental. But then if you only take a sample of, let's say, 5 individuals, you make your electrophoretic gel with 5 individuals, chances are that you are not gonna get a 50-50% at all, because let's assume that you take one. It's like tossing a coin, you know, like if you toss a coin 3 times, chances are that you get phased, you know, you get one side sometimes the 3 times, right? From which you might conclude that the probability of getting that side of a coin is 100%, but that's not what is going on. It's simply that your sample size is too small and therefore you deviate from the true parameter value. Okay, so that is the information about using a test cross. Now the other part is, how do we do it with an F1 versus F1 instead of a tester, right? In order to get an F2 instead of using a test cross. How do we extract the values? How do we extract the patterns? And what we do is, well, again, we determine which gametes can be produced by one of the F1 individuals, which is the same that we had before, is right here. And but now the other parent is also gonna have the exact contribution in terms of gametes. So we fill up our Punnett square, and some patterns start to show up. The first thing that we should notice is that instead of one horizontal line that can divide the phenotypes between resistant trees and non-resistant, it's only one of the subquadrants that can that has the resistant trees. Okay, so let's work first with this one, this subquadrant, and then identify what interactions in terms of parental versus recombinant alleles are involved. So let's identify which gametes from each parent are parental and which ones are recombinant. This one and this one are parentals, and this one and this one are recombinants. If you are unsure, again, look at how the Jefferson parent looked like. So each of these has a frequency associated to them. In the previous example, we concluded that we started with the assumption that the distance between the marker and the gene of interest was 50%. For the sake of this exercise, so that it's not as simple, we're going to assume that the frequency of the parentals is 0.8, which means that the distance in map units between the gene of interest and the molecular marker is going to be 20 map units. Okay, so the frequency of the parentals is going to be 0.8 and 0.8, and the frequency of recombinants is going to be 0.2 and 0.2. Now, some people like to use 0.4 and 0.1 instead, because the reason you are using only half of the data, right? And this is okay. You should arrive to the same conclusion. The fact is that the relationship in terms of magnitude of this value regarding this one is the same as this one regarding this one. The only difference is that one has been increased or decreased by a factor of 2, right? So this is 4 times 2. If we multiply both by 2, the relationship with magnitude is preserved. So I just look like using this 0.8 instead. It just makes more sense in terms of the map units. So we have that information there. And then the next thing that we can notice is what interactions in terms of gametes are taking part in this subquadrant. There are situations in which one of the parents inherited a parental gamete, the other parent inherited a parental gamete as well. So we get an SSLP 100, SSLP 100. Other cases where there is interaction of a parental versus recombinant, like in this one, SSLP 100 and 300, which is repeated 2 times. And the third case is that where 2 recombinant gametes were inherited from each parent, right? And there should be frequencies associated to them. The one for 2 parentals is simply going to be 0.8 times 0.8, this one times this one. The same goes for the recombinants. And for these ones here is going to be 0.8 times 0.2 multiplied by 2. There are 2 cases, this one and this one. So okay, so that's what we have in here. Here we have the contributions or the expected frequencies of each of the different type of interactions of our gametes. We have parental versus parental, parental recombinant, and then recombinant. If we multiply this by the total number of individuals that we have for each of our electrophoretic gels, like in this example, I am presenting you an electrophoretic gel that included individuals they were all resistant trees. We know that resistant trees go to the Jefferson parent, which has the molecular marker 100, so that must be the parental one, the parental gamete configuration. So we can simply multiply this value, this frequency, by the total number of individuals that we have in the well, n equals 20. These are called wells, and each one, there is one for each individual, in order to get an estimate of how many individuals will belong to that category. In this case, well, 13. These 13 right here, oops, I'm gonna erase this. These 13 right here are gonna come from the interaction of parental versus parental. Only one of them is gonna come from the interaction of recombinant, which is right here, and 6 of them are gonna come from the interaction of parental versus recombinant. So if we wanted to calculate the frequency of recombinants versus parentals, we might be tempted to simply use the parentals and parentals as direct evidence of only parental contributions. the same goes for the recombinant. And we would say, okay, there is 13 parentals versus 1 recombinant. But then what do we do with those where the contribution came from parental and recombinants? And that's where we get some issues. What I like to do is simply divide this number by 2. And if you want to look at it in here, you can see, so this is the problem, this is the representation of what we're dealing here, right? So you have an SSLP 100 and SSLP 300. From this one right here is the parental, and this one right here is the recombinant. How many of these, of the total, which is 2, there are 2 copies of the gene, of these ones, how many are recombinant? Well, it's only 1/2, right? Which is the reason why I am divided by 2. So the way that, well, by dividing by 2, I end up with 3. So there is going to be an addition of 3 parentals, an addition of 3 recombinants. Okay, and that is going to give me a total of 13 parentals plus 3 and 1 recombinant plus 3, which is going to equal to 16 and 4 parental and recombinants respectively. Okay, so by dividing this value by 4, so we end up with 4:1 ratio. Okay, so this is one way to do it. What I would recommend you to do if it starts getting a little sketchier is to, instead of counting individuals, count instead chromosomes, which is probably how our instructor told us how to do it. But not probably, that's how the instructor told us to do it, right? Is we count the individual genes, and then n equals 20, but the total number of genes in this gel is 40, right? There is 2 copies of the gene for each individual, and then we can count those at the bottom as parentals, again because that's the SSLP 100 and all individuals are res Y. So it's gonna be 23 that were here multiplied by 2, which is 46, plus 6 at the bottom, which equals 52, and then 2 here that are recombinant at the top, plus 6, which is going to be equal to 8. Okay, and that should give us a very similar value. So, well, maybe we can corroborate. So 52 divided by 2 is this correct? So, oh, excuse me. So this is 13, so multiplied by 2 is 26, and then plus 6 equals 32. So that's 32 parentals. Okay, so there is 32 parentals, and we have 2 here and 6 that is going to be recombinant. That gives us a total of 8 recombinants, which is the same relationship, right? Than 4:1. And we can corroborate that by simply dividing by 2, which is going to give us the 16 parentals and 4 recombinants, which is what we obtained previously.